I'll say two things mechanistically about this retrieval pathway. First is you have the KDEL receptor and the KDEL containing proteins like BIP in the lumens of both the ER and the Golgi, but there's only binding in the Golgi. So why is that? And the answer is pH. As it turns out, pH in luminal areas of cells tends to decline as you go from the ER out towards the plasma membrane. This is shown really nicely in this picture right here, where you can see that ER pH is 7.2. By the time you get to the cis Golgi, the pH has already dropped to 6.7, and as you move out, the pH drops down to almost 5. The lysosome, which you can think of really as sort of the end product of that sequence, is very low pH as well. That's our old friend, the V-type ATPase, going to work. But in any case, the process works based on pH, partly because the KDEL sequence, 3 of the 4 amino acid residues are charged, so it's going to be exquisitely attuned to the pH, such that you can imagine binding occurring at one pH and not at another. And that's exactly how the process works, so that you have binding in the Golgi and release in the ER, and not vice versa. And that's exactly what you want if you want a retrieval pathway that goes in that direction. And then the second thing I want to focus on is, let's say, this region right here, or this region right here of the retrograde trafficking. So in particular, I want to talk about what goes on in going from here to here to here, because there's an interesting regulatory mechanism that's important for COP1 coat formation, but also for a lot of other important physiology that goes on in the cell. And that is best talked about by zooming in on that region. So this can either be the ergic or the cis Golgi. At a region which we've already established has lower pH than the ER, such that there is specific interaction between the KDEL on something like BIP and the KDEL receptor. So the process of going from that binding to the formation of the coat involves that interaction that ultimately triggers the formation of the coat, but there's something that goes on in between in terms of the specifics of that mechanism that is important for a number of other processes. And that's going to invoke a couple of additional components that I will identify down here and then put into play in just a second. The first is the protein called ARF-GAP, and then you may recognize this one by the color code, the protein ARF itself. So let's see how they interact with each other, beginning with the binding of the KDEL to the KDEL containing protein. So that can be imagined as this, the BIP or whatever gets deposited into the lower pH environment of the Golgi, and as a result of that makes specific interactions with the KDEL receptor. That, we would say, activates the KDEL receptor, and among the things that happen as a result of that activation is the recruitment of ARF-GAP, which otherwise would be floating around in the cytoplasm on the cytoplasmic surface of the cis Golgi. But as a result of the activation of the KDEL receptor, it specifically attaches to the membrane at that region and recruits its substrate, which is ARF itself, which is in the GTP bound form at this point, but because ARF-GAP is a GTPase activating protein, it's going to activate the GTPase activity of ARF, and that triggers the subsequent steps in coat formation and ultimately an uncoating of COP1. And that's critical for that mechanism, but it's also important for other mechanisms in the sense that the activated KDEL receptor ARF-GAP complex does many other activations and regulatory events in addition to interacting with the coater, the COP1 coat. In particular, it is very important in controlling something that I mentioned earlier, the so-called unfolded protein response. And it mediates that response through regulating activities that are very, very important for a bunch of physiological processes. Those proteins are signaling protein kinases, including MAP kinase, kind of the mother of all regulatory protein kinases. But it also is important in modulating the activities of another set of our old friends, protein kinase A and protein phosphatase 1. So it's important in setting the equilibrium between those two protein kinase phosphatase activities, and in so doing can control a whole bunch of metabolic and signaling processes that go on in the cell. So that activation is very important in a whole bunch of phenomena. And then one last thing I want to point out related to this pathway is what's going on right here. So this being where it is, the surface of the Golgi, you can bet and be right and win that bet that there's going to be a trafficking motif associated with the KDEL receptor, and chances are it's going to be the KKXX motif. Sometimes you'll see associated with that particular direction of transport, the COP2 retrograde, motifs that are related to that are KXKXX, and I wanted to mention that because this is the closest analog of the slightly unorthodox trafficking motif associated with the spike protein that is the subject of assignment 2. Now, before we leave this region altogether, I want to say one more thing about the trafficking in this area at this level of resolution, or maybe two. The first being that I've talked about this KDEL retrieval pathway as if it's the only thing that is responsible for keeping BIP and things like it in and around the ER. That's not quite true. It's also the case that the job that BIP1 does in terms of binding to misfolded, unfolded proteins and maybe sending them to destruction through ERAD or not, but in any case that helps to retain proteins like that, chaperones the disulfide isomerase in the lumen of the ER. And it's also fairly common that protein-protein interactions, the formation of multimeric type proteins, is associated with the prevention of escape, so that it keeps them from leaving so that they have to be retained. And I wanted to mention that because that's going to be a part of the way in which the spike proteins and the other envelope proteins of coronaviruses accomplish what they do, which is essentially to hang out in the ergic region, so that they have hijacked the vesicular trafficking pathways in such a way that they concentrate in a region that is ultimately going to be the factory for the new virions for the coronaviruses. And that's covered in the paper that's assignment 2. And I'll go over that assignment specifically, and we'll talk about how coronaviruses do that. But it's just a matter of tweaking the trafficking signals between COP1 and COP2, and also taking advantage of the fact that protein-protein interactions can sometimes sort of override those trafficking motifs and cause proteins to stay where they're, in the case of the viruses, we don't really want them to.